de Brasil, presidente de la sección latinoamericana de la SIBI y miembro del comité científico de la SIBI. El profesor Volnei Garrafa es coordinador de la cátedra UNESCO de Bioética, presidente de la sección latinoamericana.
Professor Volnei Garrafa is president of the scientific committee of the SIBI, one of the
Debo decir que es hijo adoptivo de Brasilia por esa razón. Así que, y a continuación, el doctor Antuña es presidente del Colegio Oficial de Médicos.
I'm sorry, but the sound was very poor and we couldn't hear Professor Marcelo Palacios. Welcome to this first open debate. I'd like to thank the organizing committee for inviting the Medical Association of Asturias to represent all the Asturian doctors. Thank you, Selina, and thank you for having me here to co-chair this debate with Volnei Garrafa, who will be presenting these speakers. You know that we have 3 speakers and then 2 communications, and I hope we can have some debate afterwards for questions.
Buenos días a todos y todas.
Good morning, everyone. So this round table with the work of the conference is entitled Bioethical Aspects of Genetic Editing. As you know, the topic for this event is genetic editing with CRISPR, and this is a privilege for me and for this group of people to have such quality of speakers like the ones we're having right now. One from France, one from Spain, and then one from Canada. And then SIBI and Dr. Marcelo Palacios is always gender inclusive, so we're having 2 men and then 1 male speaker and 2 female speakers. So you're following the international standards on gender equality. So thank you. SIBI has a very special trait because aside from biotechnological issues, it takes care of the social and ecological issues. And this is a very central topic, central item of this kind of events, because here we're talking about biotechnology, biotechnoscience, and their application must be based on research and knowledge, but there is a difference with action. The German philosopher Hans Jonas talked about this, and in the principle of responsibility, because responsibility is a principle in itself, and for Jonas, the positive knowledge cannot be separated from action. That is the difficulty, and where bioethics has something to say. Of course, we can look for the truth and set it aside from the action, but from an ethical point of view, it has to go together with action. And in the conference by the previous speaker, we've known more about this. And yesterday we talked about access to the benefits of the scientific and technological development. For many authors and part of the bioethics community, bioethics is exclusively a medical bioethics. But I think you're wrong. You're wrong. Because the first line of the 7th edition of the Lovely Clark book says bioethics or biomedical ethics. For these kind of great authors, Bioethics is biomedical ethics, biotechnological and scientific bioethics. But what about the rest of the cultural differences, the diversity of cultures living in this planet? Where would they be put? What about social access?
So in 2005, UNESCO, the Organization of United Nations for Education, Science and Culture, drafted the Universal Declaration on Bioethics and Human Rights. It changed radically the international bioethics agenda. Bioethics also started to be concerned about health issues, access, equity, social topics, cultural topics. Do I want this or no? And environmental topics that nowadays are fundamental for this planet to survive in the next centuries. So it is a privilege for the SIBI and us to have at this panel the experts that we have today. The first speaker is Professor Herve Chneiweiss. Maybe he could come up to the panel, please. Professor Herve. He is a PhD in medicine, in sciences, the research director of the CNRS in France. His work is focused on biology of the population of glial cells of the nerve system, the astrocytes. His studies are focused on mechanisms that link astrocytes with the genesis and development of tumors, brain tumors, and the stem cells characteristics present in these brain tumors could develop to different therapies against cancer. So we're talking about biotechnoscience and applications. Dr. Chneiweiss was member and coordinator of the International Bioethics Committee of UNESCO for many years. As he has to travel and because of issues with the timetable, after his talk we will have some Q&A and then he can go, okay? And then the other two speakers will come along. So you have the floor.
Sorry, there is a problem with the slide, so I'm going to take my computer.
Tiene problemas con la edición.
I'm very sorry, I sent my slide, but it apparently only my CV which is on the screen, so it's not that interesting. So could you plug the... So just a minute. I'm sorry. I'm going to try to speak of these questions of governance. It's difficult to take the floor after Professor Patroneves that already made so many landmarks, and especially also after my friends Luis Montolio, who gave us yesterday at night an incredible landscape. But let's say that my first slide could have been, should have been, or will be my declaration of links of interest, because as already said by Professor Garrafa and for a while we were together at the International Bioethics Committee. I am part of several boards of international or national bioethics committees. So I will escape my slide of links of interest. And the first slide was dedicated to the fact that governance is a complex process. So Luis is going to rescue us. So what I want to mean by complexity, and this is a reference to the work we have done at the WHO expert group, and it's a privilege to be here with SIBI. It's also a privilege to be here with several of my colleagues that were at this expert group, Eva Bartnik, Françoise Baylis, and Alta Sharo. And you can refer to these documents that are on the WHO website, Governance of Human Genome Editing, published in July 2021. And we mapped these various aspects of governance. You have on one side the hard law. Hard law means international convention, and Oviedo is a typical international convention. The pity is that only 28 countries at this time signed and ratified the Oviedo Convention. You have various levels of laws, international and national laws.
How to duplicate this? How to duplicate the screen?
I don't know. I absolutely don't know. Generally, it's plugging directly, or maybe we can transfer with a key. Maybe they can copy with a key and transfer.
Okay, I have a key.
And the other aspect. And the other aspect of governance are soft law. And soft law is a huge amount of various activities that are starting from professional activities, for example, the activities that the national academies, US, UK, and Chinese develop with various summits, activities that we develop with other professionals, our society, International Society for Human Genetics, or other society like that. And I hope we are going to have slides, because at a given time, it's not only being generalities, but I'd like to present some examples of such elements of governance. I'm really sorry for this delay, fully independent of our wish.
We're saving you. We've done it. There's some. Exactly. All right. This is yours.
This is mine, but the colors are special. Well, we will see. Okay. So we skip this declaration of links of interest, but I have no share in any company and no declaration of financial links of interest. So the story is, I skip also the history of genome editing, and now we move to this aspect of hard law and soft law. And we are starting from 1215. The story started with a few reports. At the very beginning of 2015, already we had news that in several labs, especially in China, some experiments were undergone on some human embryos. And this raised the fact that several experts began to gather in January, small groups of experts. And I will discuss this difference between small groups and transparent, open discussions. And this led the INSERM Ethics Committee, and I'm the chair of the INSERM Ethics Committee, INSERM being the main health research organization in France, to produce a report. At the same time, with colleagues at UNESCO, we were also preparing an update of our reflections on human genome. And this led to discuss the application of the technology to humans in this year. And INSERM report was the first one. UNESCO report was one of the first. But genome editing, and including genome editing for health, is not limited to humans. It's also possible with animals, and I'm going to come with mosquitoes. It's also possible for plants, changing our environment, and as already said, a human being is a being living in an environment, social environment, but also the general environment, plants and climate. And so we mapped the fact that genome editing was raising a lot of opportunity, but also a lot of issues on the ethical point of view. And obviously, even if France is a great country, even if INSERM Ethics Committee is something that you are taking news of every day, I'm quite sure. We have a very lively website that you consult freely in French and in English, with more than 50 opinions now published on the website. It was not enough. And this was the start of an interesting journey all around the world, fostering an international and inclusive debate in genome editing. And this is the first step of the journey, is to say that it's not enough to be expert. It's not enough to be in such a high-level assembly. It's very important to get out and to meet the people that are really concerned and influential. And this started by gathering people at the European level, 20 organizations in 2016, with some news in journals. An important one is a kind of manifesto we published in Transgenic journal in 2017. And then we moved to various countries in Europe, Vienna. I'm going to return to this story of gene drive. Buenos Aires for the World, the International Forum on the Ethics Committee. India, returning to Europe, but going to Africa. And finally, this was the creation of this Association for Responsible Innovation in Genome Editing that Luis is presently the chair, and I am honored to be the vice chair of ARIGE. So we have the first thing that was really published was ethical issues about mosquitoes and the question of gene drive. So normally on this slide you should have had the scheme of what is gene drive, but you will not. Gene drive, to make the long story short, is the way to engineer a gene to force the transmission of the gene from one generation to the next generation. So the difference between a gene therapy and a gene drive is that the gene therapy, the new gene is going to vanish in the population because each time you have only one chance out of two to transmit the gene. With the gene drive, you force the transmission of the gene, so you are going to contaminate the population, and so you could eradicate a population at a time. And you have to know that mosquito is the worst, the most dangerous threat for human beings. Mosquito kills mosquitoes kill every year 1 million people, most of them by malaria, but you have also Zika, chikungunya, plenty of diseases that are transmitted by mosquitoes. So the question of using gene drive to eradicate the 10 population of mosquitoes out of 3,000 types of mosquitoes that are really dangerous for humans is on the air for now quite 10 years. And the questions arise to know first, is it possible? And this was the question that we put in the text that was the principle of sponsors and supporters of gene drive, first at the NASEM in the US and then published in Science 2017. First thing is obviously that we need good science, but this good science, as already said by Professor Patru-Neves, need to be for the public good. We need to promote a stewardship to know who is going to do the science, what kind of governance we need for that. And we need to demonstrate transparency and accountability. What are we going to do? How are we going to do it? And how all this is going from the lab to the open field. But it was not enough. And I think that this document is important because it was the first time we clearly pointed this. We need the engagement of community. It was already said for sickle cell anemia that the treatment is elaborated in the north, but the main population targeted is in the south. It's exactly the same for malaria. It's exactly the same if tomorrow we are using gene drive to try to mitigate or to eradicate some mosquitoes. So what kind of discussion, open discussion, what kind of engagement of communities we need to have? The stakeholders are not only from the north, are not only the people that are from Target Malaria in the UK or from the Gates Foundation. They are doing a great job. It's not the question. But we need to engage with the local communities. This is what we did going to Brazzaville and discussing with 6 national ethics committees from Central Africa. And we need to strengthen oversight and opportunities, including education, and not only education in our side, but education everywhere. So this was the first time that we put on the table the need to create toolbox. And some people now are speaking of toolbox, some other people are speaking of sandbox, but it's almost more or less the same thing. We have also scenario, and in the WHO document that we did, we have plenty of scenarios, and interesting to see, to anticipate, to forecast, because all this is about how to anticipate, how to make it flexible, how to make it responsible, how to promote science, but promote also advancement of knowledge, literacy, and engage on the long term the safety of our procedures, but also the fact that they are developing in a social environment. So this is all the goals of ARIGE, and I encourage you not only to go on the website of ARIGE, who is for free, but also to engage with ARIGE and be members of ARIGE. The cost is very little, thanks to our chair, who is maintaining a very low cost. But we need as many people engaging as possible. And you have here the principle of ARIGE. ARIGE is a very lively association with various ways to communicate. I strongly encourage you to look to the various issues of the newsletter. Some of you in this audience contributed to this letter. You have points, as I said, on the new treatments, on the impact on the environment, and so many other aspects. And we have annual meetings. We have also an Albrecht Müller Day with specific topic addressed. You find all this on the website. It's a very important place to find knowledge and the latest updated knowledge, And Luis presented yesterday the latest statement of our scientific committee. And Alta Sharo, that you will listen later this week, was seminal, and Eva Bartnik also in producing these documents. So once again, sorry for the lack of the. It seems that all the photos were skipped. But normally you should have seen on this slide a reference to the 2019 meeting in Hong Kong. Here again, I was present, and several of us who were at this meeting where Junkui He presented the genetic modification of the twins. Indeed, three girls were born with genetic modification. And it was clear, as David Baltimore, the president of the meeting, said, that it was a scientific professional failure. Why? Because the application of the technique, and I think that Luis demonstrated that clearly yesterday with all the indels and the difficulties, the technique was not safe enough, so no scientific relevance, no medical relevance, because you have many other ways to target HIV, and finally a transgression of the social environment in which any biomedical ethics develop. So I think that almost everybody all over the world condemned what did Jiankui at this time. And it was the start of another standard of governance with this group of WHO experts. And you should know that the topic that we had to cover was a matter of debate, because we decided that it should be somatic, germline, and heritable. But at a given time, we had several feedback from states that wanted to limit to only heritable human genome editing, saying that somatic, it's already business, so don't touch to somatic. We said no. We are going also to touch to the whole field for human genome editing, including somatic, including social issues. So this is why these documents are so interesting, and I strongly encourage you to go. One thing that was designed, and it's important to see, is what kind of issues. And the kind of issues, you have obviously the basic element of fairness, accountability, transparency, that are the basic elements of the European Code of Research Integrity. This is essential to build trust between scientists, to build trust between scientists and the society. But this is not enough. We absolutely need new ways to have stewardship of science, to see where the investment is going, and going in the good direction, to have adequate research capacity and technological knowledge. We need also stewardship of regulatory agency, because we are with emerging technology and they have to know how to handle them. And finally, we have to make a stewardship of scientific advance, because as Luis showed you yesterday, you have plenty of new aspects. And at a given time, it could be a different aspect. For example, yesterday, Luis presented to you the possibility, maybe in the future, to cure cystic fibrosis with this technique of base editing. But now you have small molecules, just drugs, that are able to correct the channel that is involved in cystic fibrosis. So nobody knows what is the future where science is driving us, and we have to remain always aware. And obviously we have also substantial issues, because the science is not enough. What is going to be the impact? And here again you find these basic elements of inclusiveness, fairness, social justice, non-discrimination, equal moral worth, and equal moral worth is something extremely important, with this debate we just had on dignity, solidarity, and global health equity. So we had general recommendations, but also practical recommendations. And among these practical recommendations, we have now registries. And this is another kind of governance. Registries are ways to have a transparent view on who is doing what. It's like more or less like the clinical trial registry where we see all what is happening. We have also recommendation on how to prevent medical travel, ethical dumping, and to promote education, engagement, and empowerment. And I am going to continue my presentation on these elements of education, engagement, and empowerment. UNESCO at the same time was organizing a certain number of roundtables. You can find them on the site, on the website of UNESCO, trying to engage this question and to improve the literacy of the general public on these questions. Not only on the question of human genome editing, but also the impact on the environment. You have all edited plants now. You have edited animals, not only mosquitoes, but also cows, for example, to make cows without horn. Other animals, including recreational, creating very small pigs that are pets like very small or very muscle dogs. So you have plenty of issues, and I strongly encourage you to see all these roundtables were captured, and you have movies, and they are going from questions of science toward question of how to engage the community. The roundtable with Kevin Esfeld, for example, on GeneRive, how he engaged the dialogue with Maoris in the New Zealand or with the Nantucket people is really of great interest. So it's not enough to make good science in the lab, it's important to get out the lab and go to see the people. And this was also the case of trying to animate the Article 28 of Oviedo. Many people, especially for genome editing, are focusing on Article 13, because Article 13 is the one who said that any genetic modification that should be done in the purpose of transmission to the next generation, so heritable genome editing, is forbidden. But very few people are focusing on 28, and 28 is that any kind of new development in the biomedical field need to be under the scrutiny of the public debate. You absolutely need to foster a public debate on any kind of new innovation in the field of medicine. And so after the Hong Kong meeting, with a very famous filmmaker from Australia, we created an international collaboration where at the beginning 15 countries were involved. At the end, only 5 countries participated really in the consultation to engage with the public, with the people. And this was a Global Citizen Assembly. You can find the purpose of the Global Citizen Assembly either on the website or in a paper that we published in 2020 in Science. And as you see, it's a rather long and difficult process, because you need to have the people that are engaged to come for 5 days to learn more about genome editing first, not to discuss in the vague at large, but to discuss with real knowledge of what is the science, what are the issues. And then you have a debate, and finally they are giving us conclusion. And our idea, but it was before the COVID, it was before the Ukraine war, it was before the Israel-Gaza war, our idea was to convene at the UN for the 25th anniversary of UN and to present to the General Secretary the result of the Global Citizen Assembly. We don't know if it will be possible any day. So here, to illustrate the fact, normally you should have also the number, that it was all over France, 300 people gathered, and a lot of young people. At the end, only 184 completed the whole survey, but it was already something to do. So, everything is missing. No. The very interesting thing I'd like to show you here, I hope I remain in the time. Yes, I'm close to the end. The very interesting thing is that the people are very open to genome editing, especially when it's a matter of cure. They want supervision. They want experts to be involved in the supervision. They don't want to have some kind of Soviets. They want really that the professionals are involved in promoting, but also surveilling, and they want a clear legal frame. What they don't want discrimination. They don't want to use this emerging technique, very expensive, if we have another way to address the question. They don't want heritable genome editing. This is the question of future generation, but I will not go completely in your direction. What we are doing today with the climate change is our decision, and we are engaging the future generation with our foolishness. So we are doing something very wrong for future generation because we are not acting for climate change today. So we are already doing something without any kind of genome editing. So call to our responsibility, so what we are deciding for future generation. They also, very interestingly, I recall you that we had a lot of young people in this audience. They don't want a loss of diversity. They want humanity to be diverse. They don't want to homogenize everything by deciding what is normal and a standardization. And they don't want aesthetic modification. And plenty of questions. They say, okay, we need to discuss what kind of disease is eligible, and we should try to make a kind of disease. What should be an abuse? What kind of consent? Because all this is very difficult question to understand. So how are you going to really inform the people about the consent? What will be the technical control, level national or international level? Should it be prioritized to a certain number of disease, but also disabilities? And what mean enhancement? But it was already said before. And so the main question today, all of us, we say the same thing, but it's needed to tell once again. The question is access. Access is the price, but access is also the infrastructure, because as far as we are using ex vivo treatments, we need to take the cells from the individuals, to change the genes, to select the correct corrections, to amplify the cell, and to make a new infusion, all this in GMP conditions.
Excuse me, Dr. Herve, there are 2 minutes.
It's my last slide.
Okay.
For example, a proposal that was made by the French National Academy of Medicine is to create national infrastructure and maybe to promote open label, as it was done already for some essential drug for HIV, as it was done also at a given time for gene patenting, especially the case of the myriad trying to capture BRCA1, BRCA2 test. And I am concluding on this. Thank you so much, and sorry for the technical drawback.
Thank you very much. And in this moment, there are open by questions and discussion. Están abiertas entonces las preguntas. La primera del doctor Montoliu. ¿Sí?
Hola. So thank you, Herve, for this presentation and for all the topics that you presented. The question goes for both of you, for Professor Patros and for you, Herve. And you have presented the need for involving the society, involving the people. And you presented the initiatives you've done at the INSERM, the initiatives that were carried out by RICH, and also the Global Citizens Assembly, etc. My question is simple. Are we doing enough for public engagement? What else can be done? Because if we are not reaching the society, we will have a problem, not only about understanding, but also about justifying and just letting them know and hoping that they will understand when we will be presenting these different therapies. So what else can be done for public engagement?
Thank you, Luis, for this question. Obviously, we do what we can, but what we can is something. First, it needs to be diverse, and the basic things is to have the general way now of open science, which is making the data, making the science available for who wants to go to know the science. So this is what our institutions, I'm sure in Madrid you have a website with a lot of data available. This is what Cochrane conferences are doing to make meta-analysis and promote it, TEDx and things like that. But it's not enough in the way that people have their daily life. They are not concerned with, when you speak of genome editing in the street, they have the questions of eating every day. They have the question of making their kid growing well, environment, things like that. They cannot be bothered with science every time. So we have to go and meet the people. It's a need to make a day of science or a week of science, to go in supermarkets, to go in the open field and be ready to discuss with the public. It's also the need to go in the various countries that are going to use the techniques. And we have seen with COVID how a treatment that was costing only a few euros was difficult to raise and go to the countries that needed it. The failure of COVAX initiative in Africa was something terrible, because you should know that now the general vaccination against tuberculosis, against the other disease, dropped down by 30 to 50% because of the distrust against vaccine, because of the post-COVID story, because they couldn't access the treatment. So we absolutely have to regain this trust, and to regain this trust we have to go and see the people. We have to follow some examples. It's not because Garrafa is here, but Brazil opened the field in 1997 when they decided to make HIV treatments off-label, and it was an incredible story with the fight with the industry in Johannesburg trials. But we have to see these examples, and we have to move out of our labs. This is what we are doing today, to discuss multidisciplinary. This is what we are going today. But outside of expert meeting, to really meet the public where the public is.
Siguen abiertas las preguntas, por favor.
More questions.
Es importante registrar, según la organización.
According to the WHO, we need to know the net price of a vaccine, because it costs 87 dollar cents. That's the net price, or the net cost. Imagine you could do that vaccine for free or for its price, for its cost, to the 11 billion people living in the world. But even when this has been proposed, 14 countries voted against it in Qatar, and then it led to the non-universalization of vaccines. But imagine just 87 cents, or even in the case of more expensive vaccines, $4. Multiply 8 billion people times $4. It is very affordable. Any country could pay for it. Any big country could pay for it. So the problem is not the cost. The problem is the lack of political will. Because in Doha, in Qatar, in 2001, 14 countries voted against this. And just because 14 countries voted against it, there was no unanimity and no universal vaccination is available, just because of 14 countries. Of course, because of the international pharmaceuticals lobbies. So yes, in Africa, less than 35% of the people are vaccinated. And because virus travel long distances very quickly, without universal vaccination, without universal immunity, they will continue to spread. So we need the political will that we saw in Brazil a few years ago with HIV. In Brazil, there's dengue. Many people died of dengue. So Brazil really has made the decision to treat its people in the most universal way. Thank you, Doctor, for your excellent presentation.
Presentar a doctora Moratalla, por favor.
No, tú yo presento las dos comunicaciones del film.
Okay. Nuestra próxima conferencia.
Our next speaker is Dr. Natalia López Moratalla, a very known person in Spain, a great researcher. Just like our previous speaker, Dr. Chneiweiss, Dr. Moratalla has a chemical science degree from the University of Granada. She has a PhD in physiological chemistry from the Complutense University, and has been a professor at the University of Barcelona, at the University of Valencia, at the University of Navarra. Is a honoris causa doctor for the Pan American University, and an emeritus professor of biochemistry and molecular biology. She's also been the honorary president of the AEBI. Well, I'm going to talk about what genetic editing cannot modify in human beings. So genetic editing is a rich array of techniques that are always evolving to modify the DNA in living organisms with a specific purpose. We have many different tools, as Dr. Montague explained yesterday. So in our collective imagination, modification of the DNA is associated with a lot of hope or fright, because we think it's going to change our nature. So I'm going to talk about my reflection on the ethical principle of the social information that we need to inform the society about these and future evolutions. Because scientific questions, just like biological questions about body and the human brain, can always be replied to, because we can use our scientific evidence to give it an answer at short or long term. So we can reply to those questions, we can answer those questions based on the technology that enables us to get to necessary data for that. But human biology is not just simple zoology. There is a reality aspect in each person that does not depend on their genes and the regulation. So there are many human questions, human issues that cannot be solved through the scientific method. We cannot answer them just with science. We have two levels that are intrinsically merged in our body. We have the biological level, just like animals, but we also have relationship information, relationship of communication with the rest of the people. So we need freedom, which is not a biological property, because it means getting out of the cage of the animal automatisms. So yes, it is possible to replace amino acids in a protein by editing the DNA of the codifying gene. It's already in place, as you have seen. We've seen clinical essays and papers that are looking for that therapy. It is very difficult, and it starts with the blood cells that can be taken out of the body and reproduced, and then in the tissues such as the heart, and then the brain maybe later. Monogenic diseases are easier to treat than the multigenic. We can also change the amount of a protein to create an epigenetic modifications, although it has some difficulties because the signals, the regulation basically lies in the sponsors of the DNAs through a sequence that have methylated cytosines. Epigenetic rejuvenation offers possibilities to improve human health and protect our life expectancy, not just to get a longer life, but to enhance or optimize our biology. For example, some physical exercises regulate genes by producing molecules that enhance or optimize the gene functions, and in the near future those genes can be edited. But at the same time, I don't think we can lengthen our life a little bit more. We cannot make life longer because there is a system called cell proliferation control. used to control cancer or used by cancer. And we also have some more difficulties. I'm going to talk about 4 difficulties. The first one is the great epigenetic regulation that the brain has for a gene. I'm talking about the brain. The difficulties in editing the genes and the epigenetic modifications of the brain. The first one because of the great epigenetic regulation of the brain. Secondly, because of the cell complexity of the brain regions. So we need to know more about the progress in neuroscience. October the 13th last year, we learned that we have over 3,000 different cell types. The proportion of types of cells according to the genes expressed is individual. Is person per person. So that's one more difficulty. And then we also have, in the third place, structural complexity. From the beginning of the embryos, we have lots of interconnections and complexities, intercellular, interneural complexities, and then in the teenage years, we get some kind of final structure. But look at these connections between the neurons, and this is like a network that creates several groups and cell areas in layers to give rise to an unlimited brain. These structures have lots of plasticity and lots of connections, and it all stays in the brain. There are no such thing as 2 identical brains. So this is ever more complicated. We have 2 traits that depend on a gene or several genes that could be edited over time. One is the analytical intelligence. We know that it depends on what you got from your parents. For example, some children are experts in the 3 basic functions at age 6, and they will be, as adults, knowledgeable and proficient in all those 3 structures, and they can have a very high intelligence quotient. We know there is a correlation between analytical intelligence in both reading, writing, and arithmetics. And there is, as I was saying, this correlation between the innate analytical intelligence and the maturity or the maturation speed of the frontal lobe region in the left hemisphere in the childhood and the teenage years. On the other hand, we must know that temperament is innate. For example, kindness depends on the cortex, the social brain or neuroticism, our conscience, awareness, executive control, and impulses. So that all drives the character, and influencing this would be easier than gene editing. So in the future, yes, there are things that depend on genes. It is true that some traits depend on genes, but we cannot modify them through genetic editing, even if it could be made possible in the future. It is better and advisable to influence those traits with other methods. So as I was saying, we have a biological level and a relationship level, but for that we need freedom. And in the brain we can find a physical space, which is the brain itself, the physical brain, that in parallel creates a mental state. For example, we can have mental representations, we can also have mental patterns, but we have something else, as I was saying, the relationship level, and any changes in that will create sequences of mental states. But for that we need some working mental space which is timeless, unlike physical space. For example, we have self-awareness, we have an activity pattern, a mental state, neural networks, but then we have some self-awareness. And finally, we have the fourth factor I was referring to, the functional complexity of brain. We have the axons, the circuits, the networks, the change over time that can be trained for new properties that sustain this network, changes in the layout and the configuration and the setup of the networks. And brain is never off, is never completely off or on. This is very important because there is a direct activation of it and a different array of mental states depending on the setup of a specific task or a need for a specific execution. So yes, we have a physical brain, which is an organ that we need, but not alone, because of course we need the activation of the brain done by the subjects, and that mental working space is not a physical space in the brain composed of the functional networks, but those functional networks are necessary. They need to be active, and we also need specific regions that also need to be active because they sustain the networks. So it's not stimuli that will govern the answers or automatisms. We need something else. And cognition and all human faculties, all human skills require two abilities that do not directly depend on the genes. We have human exclusive timeless memory that shares some brain regions that enables us to remember the past and the present, and we make plans for the future. That timeless memory underlies the central network of the brain that connects the forehead with the upper parietal region, so that mental time is subjective. It does not rely on the genes. Past and future do not exist in reality, but only in the reference of the subject. The second thing I wanted to talk is about the need for us to get out of the automatisms, to get rid of the automatisms. We can stop the stimulus from the amygdala to the orbital frontal cortex. We can stop it. I can say, stop this, stop thinking about this, stop reacting to that stimulus, or I'm changing my reaction. I don't want to do this. I don't want to follow my spontaneous or automatic instincts. I can stop it, and I want to stop that answer to that stimulus and instead decide what to do. So stop and think. That capacity gives us freedom. It's a personal freedom, a personal stop. But the human brain is full of chaos, and it has its own dynamic. So when for example, when the initial characteristics change, the trajectory changes too. It can convert into a mental or brain stability point. But it is not determined by genes. we can break the automatisms, and this is all driven by the habits and the relationships that person has. So it is self-determined and free. So by way of a summary, I will say that the properties or characteristics that depend on a gene or several genes can be genetically edited, because as we can see, this method is evolving. We can replace a fragment that has been altered, as we saw yesterday, and over time we will see more and more possibilities for those therapies and methods to replace genes. But epigenetic modifications through genetic editing could change some physical traits in the body by up-regulating or down-regulating specific gene product. So the brain thing, even if with the serious difficulties, given the complexity of the brain itself, can still be genetically edited, especially in the field of neuroscience and the genetic editing therapies. Okay, that's true. But then we have the mental aspect that requires us to have some specific neural networks on. So for that network to be on, that doesn't depend on the genes, but an on-time or an occasional activation. So genetic editing doesn't have a say in here, has nothing to do here, because this is not due to a scientific question. So we cannot anticipate an answer to that question with the progress of the knowledge on the brain and genetic technology, because it is beyond that. The mental work that enables us to think, know, make decisions cannot be genetically edited, and the human genuine capacities cannot be enhanced through genetic editing. And that's it. Thank you.
Tiene más 7 minutos.
Okay. You finished? Thank you so much.
Para el debate.
Sí, ya se queda. Sí. Muchas gracias, doctora Moratalla, y también por el tiempo que nos ayudó bastante a quedarnos justos. La tercera y última conferencia de esa magnífica mesa redonda que tenemos aquí, proporcionada por SIBI, será la doctora Françoise Baylis. La doctora Françoise Baylis, muchas gracias, es profesora emérita de investigación de la Universidad d'Alousie de Canadá, miembro del Comité de Planificación de la primera y de la tercera cumbres internacionales sobre edición genética de 2015 y 2023, miembro del Comité Asesor de Expertos de la Organización Mundial de la Salud sobre el desarrollo de estándares globales para la gobernanza y la supervisión de la edición del genoma humano de 2019 a 2021, y también miembro de la Junta de Gobierno del Consejo Científico Internacional de Canadá y vicepresidente de su Comité para la Libertad y la Responsabilidad de la Ciencia. Muchas gracias, doctora. Está con la palabra.
Gracias. It's a great pleasure for me to be here today, and while I can understand Spanish when people speak to me very slowly, I would not attempt to give this presentation in Spanish. I'm just going to... Okay, great. So the first thing I want to do is set the stage for you. And what I'm trying to do in this presentation is to invite you to consider a question that I think is actually extremely important to the whole discussion and debate about heritable human genome editing. So you've already heard from Herve's presentation that we worked together on the WHO report, and in that context we looked at somatic genome editing, germline genome editing, and heritable genome editing, meaning with the intent of pursuing a reproductive project. And so the question I'm inviting you to think about today is whether reproduction is and should remain a personal decision, or whether there's a scenario in which it's reasonable to believe and to defend the belief that in fact, in some sense, it's not a personal choice. So what I'm asking is, when can we, if ever, when should we, if ever, infringe on personal liberties for collective benefit? And so I have here two images where, in fact, many of us have argued that collective interest should override personal choices. So in the context of climate, we're saying maybe you don't have the right to keep polluting because all of us will bear the consequences, and we have communal obligations which will have us constrain your liberties. We certainly had that kind of debate as well around COVID, and you've already heard how there was pushback in some sense from several different kinds of communities about whether communal or collective interests could override personal goals and objectives. And so I'm saying to you today that we, in the broadest sense of we, are on the cusp of potentially making significant changes about the ways in which humans can reproduce. And the question, and my original title was Infinity and Beyond, meaning it's potentially limitless what we could do. My question is, how do we think about this activity, and does it belong in any place other than in the private realm of reproduction? And so we might have questions about why and when that remain personal, but does the question of how we reproduce leave the personal domain and go into the collective domain? That's the question I have for you to consider. The structure of my presentation is going to be in 4 parts. First of all, I'm going to say what are designers' babies, because I want to make sure we're talking about the same thing. I'm going to then specifically ask the question about CRISPR babies. Do we want them? Maybe not today, but maybe at some future time. What comes next? And I'll just give you a brief aperçu of what people are thinking about in terms of synthetic biology. And then the last pivotal question is, what kind of world do we want to live in? And is it one of unconstrained freedom, or one in which freedoms are constrained? And if so, in which ways? So designer babies, what are they? Well, when we talk about designer babies, we very often make reference to Louise Brown, the first person born of in vitro fertilization. And this is clearly an important event because it makes the human genetic material available outside of the human body such that everything that follows in terms of manipulations becomes possible. But the point I want to make is that despite the fact that this is a very important innovation in terms of assisted reproduction, Louise Brown is not a designer baby. And she's not a designer baby because if you look at the technology, all that you did is circumvent a natural process. You didn't actually design anything, right? And so what you have with that intervention is you're removing the genetic material from the body. You're making the egg and the sperm meet in a petri dish. Hopefully there's fertilization, and then you're transferring the fertilized ova back into the body. There's no design question. You are doing an end run around a particular kind of physical barrier or other kinds of causes of infertility, and the gamut of reasons for using this technology has changed, but I'm arguing you're not yet doing design. The first designer baby, I would argue, happens with mitochondrial donation. And in that context, the reason I think about this as a designer baby is because something is happening that could not have happened but for human intervention in a particular way that has an impact on the genetic material. And so this technology is somewhat different in that you're starting with 2 eggs, and there's the belief that there's a problem with the mitochondria. So if we think about the chicken egg, there's a problem in the white of the egg. And so what you're doing is you're removing the nuclear DNA, the yolk, the yellow, and you're taking that and putting it back into a different egg where you believe the mitochondrial DNA is healthy, and then you go on to have fertilization and a transfer and hopefully a birth. In this context, we've talked about this as the three-parent baby, and that's because in theory you have genetic material from three individuals: the woman who provided the mitochondrial DNA, the egg white, the woman who provided the nuclear DNA, the egg yellow, and the male who provided the sperm. This is not something that would have happened in nature, but for a particular kind of human intervention. Another kind of designer baby, we've also had references to this in several of the presentations thus far, is by Jean-Q Hay, who in 2018 announced to the world that he had made genetic modifications using CRISPR technology, arguably in order to avoid the risk of HIV in the offspring. And I'm not going to talk in any great length about whether what he did was right, wrong, or successful or not from a science point of view, but I am going to put on the table, here's another possibility, perhaps perfected at a future time, of reproducing in order to achieve a particular kind of goal. And again, the way in which the genetic material would be used or manipulated would not be possible but for the human hand. So in this case, what you have is you have fertilization taking place outside of the body, so the same in vitro fertilization, but you then have the genetic modification happening on that fertilized egg. The embryo develops, it's implanted, and hopefully a child is born according to the technology. We've also had images to do, again, going back yesterday to Luis's presentation about the ways in which we can do the genome editing. Here you have samples, just a cartoon really, showing you what can happen in terms of deletions or in terms of additions. But as the point was made, in many cases it's just going to be disruptions, and the DNA will mend itself and changes will take place. So we understand that CRISPR babies then are designer babies. And I want to ask the question in the second part of my presentation, do we want them? Do we want to have more CRISPR babies? We know that we have 3. We could, in theory, have more as the technology were perfected. Well, one of the things that many people talk about in this context is that it seems to support a certain kind of liberal eugenics. And in this case, people are asking the question, should the fertility industry be allowed to continue to pursue whatever goals and objectives unconstrained? Are these kinds of reproductive choices truly personal, private choices? We have lots of conversation about the elimination of disability, and then conversation about what constitutes a disability. And is a disability any more than an unwanted trait, and then unwanted by whom? By the individual, by the family, by the community, by society writ large, which sees itself bearing the consequences, whether that's in terms of providing financial resources, modifying environments, etc. But the one point I wanted to make with these particular images is to show you what appears to be the inherent desirability of whiteness. And I think we need to pay attention to that, especially in the context of things that we've raised already with respect to non-discrimination, with respect to equity, with respect to justice. What are these images showing us but what some people believe to be presumably an ideal future human? And so I think we need to think about the images that we portray when we try to put this forward as either something to pursue or something to be fearful of. I want to make it clear that if we look to the public, we have seen a number of efforts in different communities, different times, relatively modest, stating that they object to this kind of genetic modification. This image was taken at the time of the most recent international summit in London. But there have been others in other countries with a similar kind of pushback, saying we don't want the equivalent of human GMOs. So one of the things that becomes important to think about then is how do we do appropriate, interesting, useful, informed public consultation? How do we respond to this kind of pushback? The other thing I want to put before you is the fact that despite what any kind of members of civil society might be doing, we have to look at other areas in which there's governance in some sense, and this is a mixture of both hard law and soft law. If we look at the language that Herve gave us in an earlier presentation, meaning this map here reflects both regulations, guidelines, etc. And if we look specifically at human genome editing for the purpose of reproduction, what you can see in this place is that there's absolutely no country that has a piece of legislation explicitly permitting this. There are countries which have nothing, which means in theory one could pursue this goal or objective there. You have places where when you look at the legislation or the guidelines, it's actually very difficult to determine what it is that they're saying in this space. But the one thing that isn't there and could be colored in blue is international waters, where you could have a very fancy boat and set up your lab and do anything. But I think what's important to think about is what is this suggesting in terms of support for heritable genome editing for the purpose of reproduction. It's suggesting that at the present time, this is not something that any one country is actively advancing. Now, what's important is that that's not true if we just look at germline genome editing. So going back to that distinction that was made, germline genome editing is doing that same work in terms of manipulating the human embryo, but that work remains in the lab and has a number of discrete goals and objectives having to do with better understanding human development, improving reproductive technologies, perhaps developing better abortifacients, better understanding miscarriage, etc. But that is work happening in the lab with no intention of a transfer to a woman for a reproductive purpose. And here you see a very different map. So you see a number of countries where they clearly do have legislation or guidelines in place saying that this is perfectly acceptable, but you do have other places where that's not the case. So one of the things to pay attention to is how are these maps going to evolve? What colors will change, and will they change differently for the two different technologies? Now, the one thing I wanted to make sure that I did, and I know a number of other people have done this, but I do think it's important, is here we have two women at the center of the science who were awarded a Nobel Prize for this work. And I just think it's really important to understand how quickly we moved from their work to ethical controversy. So early on, the papers are published in 2012, and the first kind of concern that we have in a very public way is in 2015. And it's research done by this Chinese scientist, Huang, who basically reports, meaning publishes, a paper in which he says he has done human germline, so work in the human embryo, using non-viable human embryos, so embryos that would not have been transferred for a reproductive purpose, but it has taken these non-viable embryos and tried to manipulate them. There's no transfer to a woman, and it's by and large not very successful. He tries to publish this in Nature and in Science. It's not accepted, and so in either of these two journals, and so in the end it's published in Protein and Cell. Much happens in that year, and it culminates with the first International Summit on Human Genome Editing. And I was a member of the planning committee for that, and that's just by way of transparency. So in December 2015, we had the first International Summit in Washington, D.C., and in very simple terms, there's strong approval for basic research happening in the area of human genome editing. There's approval for clinical research using somatic genome editing for the treatment of individual patients who are alive and experiencing health problems. But then with respect to heritable human genome editing, the statement is very clear, and it basically offers up an ethical framework that has only two conditions. And one of the things that I say all of the time is that this is extremely exceptional. It is very rare to have an ethics framework that only has two elements. Element number one having to do with safety and efficacy. Element number two having to do with broad societal consensus. And when I'm being facetious and I tell you, on the one hand, it's a great ethics framework because it's simple, it's straightforward, it also is extremely useful because nobody really knows what it means. And I think what's important to appreciate that is that that's true both with respect to the first statement, which may sound robust and may sound objective, safety and efficacy, but safety and efficacy according to whom and at what standard? It's never going to be 100% safe and efficacious before we do it, so is 90% good enough, 70%? It just have to be 50% plus one. Well, that tells you already it's value-laden, about which we have to have much discussion and debate. And the same is true with respect to broad societal consensus. What does it mean? How do we know when we've achieved it? So I want you to take away that, from my point of view, we have this amazingly great ethics framework, and part of the reason it's great is because nobody knows what it means, so we're going to have to do work, important ethical work, to get from this kind of statement to how you might operationalize it. I've done a lot of that work in terms of my own contribution to the academic literature, and I'll return to that in a minute. Fast forward, we're now in 2018. You've already heard much about Jean-Q Hay, and the difference here is that in 2015, science happens and we have a meeting. In 2018, a meeting is planned, and on the eve of that meeting, we have the announcement that a particular scientific practice has happened, and the world learns that at least 2 babies at that time have been born. And here you see the statement from the organizing committee that time, and they say here that germline genome editing could become acceptable in the future if the risks are addressed and if a number of other criteria are met. And the claim is made that now is the time to define a rigorous, responsible translational pathway towards such trials. Now, I was not at that meeting or on the planning committee, but one of the things that I thought was interesting is I, like many others, was watching this, and in my time zone, very late at night or the early morning, and I hadn't anticipated that this would be the final outcome. And the only reason I say that is that Feng Shuang, one of the scientists in Harvard, when he was asked for a quote once this became known, and he was getting on the plane and he issued a written quote, he actually quoted from the 2015 statement. And the same is true of David Baltimore, who actually chaired both of these meetings. In the initial moment, he said, speaking for himself, almost exactly what had been said coming out of 2015. So in 2015, we have somebody doing research, non-viable embryos, not transferred to a human, and the statement is, don't do this unless you can meet these 2 conditions. 3 years later, we have somebody who's done it, and the statement isn't, don't do this. The statement is, well, people are going to do this in some sense, so we better have some clarity with respect to the context within which that might or might not happen. The elements of this rigorous, responsible pathway forward are these, according to some of the writings there. And I'm not going to go through each of them in the interest of time, but I do just want to talk about one of them very briefly, which is this notion of compelling medical need. And this is but one example. One example where you might have a compelling medical need is a situation whereby you have a couple that wishes to have children that are both genetically related to them and healthy. And there is no available technology other than heritable human genome editing that could respond to this desire. There's nothing else that can be done to respond to the desire of having healthy, genetically related children. Now, I'm going to say to you, and it's not today's talk, I don't think that that's a good enough reason to go forward with this technology when I think about all of the opportunity costs in terms of the investment. But that's not today's talk. All I want to do here is just highlight one of the opportunity costs if we take the example of cystic fibrosis. And these are numbers generated by an IVF clinic in the United States using the data and the numbers for the United States, saying that if we took cystic fibrosis as the example for which we wanted to respond to the needs of a couple that could not otherwise have a healthy, genetically related child, we'd be looking at 1 case every 15 years. The opportunity costs of this technology are huge, and I'm not saying they couldn't be met, but we'd have to understand the context in which we're arguing for them. Very briefly, we skip ahead to 2019, and a number of us who were part of 2015 and were concerned about the direction of this science called for a moratorium. I mention this now because I think it's really important to put it into context, because I have heard a number of people at different meetings over the past few years suggest that we have a moratorium. And we don't. I want to be really clear about that. You have a paper that was written by a small group of people calling for a moratorium, but there is no mechanism, no soft governance through which any kind of moratorium has happened. It's just happenstance that we don't yet have other babies that we know of that have been born in this way, but it's not because there's a moratorium that's being respected. There is no such moratorium at this time. There has been the 3rd International Summit, which I've already alluded to, and one of the things that I think is important here is that you're seeing, I think, a little bit of a shift, saying that we still need to ask important questions about whether or not we should use this technology in this context, and the point is made that we have not yet had the societal discussion and the policy debates that we need to have. I'm going to quickly try to summarize for you where I think that policy debate happens. So in 2015, it would be irresponsible. In 2017, and this is a NASM report, it should be permitted, but for compelling reasons and under strict oversight. 2018, the Second International Summit, time to define a rigorous, responsible translational pathway forward. And in 2023, a pulling back. Public discussions and policy debates continue and are important for resolving whether, and I think that's an important word, whether, not when, whether this technology should be used. I only have about 5 minutes left, so I'm going to race through the last few slides. What comes next? That's a reference to synthetic biology and returning back to the theme of my presentation, to infinity and beyond, the limitless possibilities I'm thinking about. And here what we have is the possibility of in vitro gametogenesis, where we'd be making custom human eggs and sperm in the laboratory from any cell in a person's body. And I often joke and think, well, if I wanted to have anyone's child in this room, I just make sure I have coffee with them, and I take the coffee cup with me, and I try to get somebody to extract some DNA, of course, in some future world, and I would have their baby, and it wouldn't matter whether they were male or female, because I would just have them make sperm from that somatic material. A little bit of science fiction, but again, what becomes important is thinking, what's the context in which we will constrain or encourage this? And this is a similar kind of technology, but starting with stem cells. So what we have, and this is in the last year and a half or so making headlines, is groundbreaking synthetic human embryos being made without eggs and sperm. What kind of world are we going to move towards? What kind of reproduction do we think is important? This has already made its way into the Parliament, and it's being argued for on the following grounds. Why would we want to do this? Why would we want to make human embryos from stem cells? Well, we'll better understand basic human biology. We'll deal with problems like early pregnancy loss, help with IVF outcomes. We can do disease modeling, and the list goes on. The point here is that they're making very clear what are some of the benefits. What I think is interesting is not these benefits, but the benefits that other people see and point to. This would be a way of making gametes for people who don't have any gametes. For example, people with Turner syndrome, people who are undergoing cancer treatments, etc. So there are women without eggs, we could make some for them. There are men without sperm, we could make some for them. It's also seen as perhaps being important for allowing same-sex couples to have genetically related children where both of them would have made a genetic contribution. It's also seen as a possibility for solo reproduction. It's another possibility in order to address the need for, desire for, genetic material in the lab, because you just make eggs, and that's be a whole lot easier than extracting them from women. I think the thing, though, that becomes important isn't so much the why, in some sense, but the what. And here I'm just going to allude to the fact that one of the things that I find quite fascinating is the debate we're having now about what we're going to call these organisms. Are they really synthetic embryos, or are they something else? And I'm not going to go through the naming debate. It's just to say the naming debate is actually about ethics, because there's an effort to make sure that whatever happens here is not constrained by the current limit on human embryos, which is the 14-day rule. And many of you may have read that there are places where they're now saying explicitly that extrauterine embryos are in fact extrauterine children, and for many people that's quite concerning. So I'm going to end with the provocative question, or thought-provoking question: what kind of world do we want to live in terms of what that next generation is going to look like? I've been arguing that this is an important question to be answered by all of us, not in the lab, not in the boardroom, but in the kitchen, because that's where we all have something in common. The answers to some of that from my limited perspective are in my book, Altered Inheritance, so here's a little bit of promotion. If you find anything that I've said here interesting, it's not in the book. No. What's in the book is actually the effort to unpack the ethical arguments in support of this technology and the ethical arguments against this technology, all against a belief and a framework that what's important is consensus building. And so in some sense, I'm stuck in the mud. I'm still back in 2015. I'm still arguing that we ought to work towards broad societal consensus. I understand not everybody agrees with me. Not everybody thinks it's possible. And my belief is we will be better off for having tried, even if we never achieve consensus. Thank you.